Welcome to our webinar entitled A Fork in the Road, Assessing Diversity in Early Phase Clinical Trials in a Rapidly Changing Regulatory Environment. Today, we're being sponsored by Biogen and Altasciences. I'd like to welcome Sean Ratcliffe-Smethurst, PhD, Senior VP of Head in Quantitative Sciences and Development Operations at Biogen, and myself, Beatrice Setnick, Chief Scientific Officer at Altasciences. During today's webinar, we will be discussing strategies that proactively address diversity and representation in our early phase clinical trials, particularly in a challenging and changing regulatory environment. We'll also be discussing the benefits of conducting pharmacogenomic drug-drug interaction studies and other key NDA-enabling studies in early drug development. We'll be considering different strategies for engaging, recruiting, and retaining diverse study volunteers in early phase to fulfill diversity requirements, and discussing the integration of early phase data to support a robust clinical development program with a diverse study population representation. Now, Sean, I'll give you the dialogue.
Thank you, Beatrice, and thank you for the introduction to this really important webinar. And I'll give a little bit of a scene set as to why now and why this is important. And we'll start with the draft guidance on diversity action plans that was issued by FDA in June 2024. This guidance summarizes the need to have specific action plans in place to improve enrollment of participants from underrepresented populations because there's been a historical lack of diversity in clinical trials. And it's also important thereby to understand the impact of drug efficacy and safety across different populations to be representative to the ultimate patient population that we serve with our medicines. This diversity action plan guidance also shows FDA's commitment to advancing health equity. This draft guidance has had a little bit of an update since the recent administration shifts and changes in the US. So the guidance document had been removed, but has been reinstated as of February 2025 following a federal court order. So if we move to the next slide. So why does clinical research need to sort of incorporate diverse patients? Historically, clinical research has not served all communities or represented all communities. And therefore, clinical research should really reflect the growing social, racial, and ethnic diversity of the US population. This guidance document highlights the importance of doing so. It also highlights that this diversity plan will be important to have in place for late-stage development programs, and we'll go into the specifics of the timing of that a bit later on. Importantly, some of the concepts that are outlined here, that there's equitable access to clinical research, and that is impartial and fair access to clinical trials, and so that we don't get divides based on representation or communities that are served differently. We try and encompass both underrepresented and underserved populations, but also the inclusion of the experience of being in clinical research, that all participants are welcomed, valued, and can fully participate in clinical trials. If we move to some of the key guidance elements, we talked moments ago around the requirement for a diversity action plan. The diversity action plan in the draft guidance is required at the time of a phase 3 pivotal study protocol being submitted. And there are recommendations throughout the guidance that sponsors don't simply think about diversity at that point in time, but they think about it all the way through the life cycle and their development plan in their design of studies. The components or the goals, let's move to the goals first. The goals of diversity action plans to ensure adequate representation of racial and ethnic minorities in clinical research so that we can improve the generalizability of clinical trial results, as well as enhance safety and efficacy assessment across different patient populations. As such, the components of the DAP, which we're shorthand calling the Diversity Action Plan here, is to start at the top with enrollment goals, to really set enrollment goals based on demographic data for the disease area under study, to employ recruitment and retention strategies that really address those diverse populations, ensure that we can bring trials to patients and meet patients where they are, to provide a justification for those enrollment targets and strategies. So leveraging epidemiology data to really understand the patient population under study, the different populations and subpopulations within a particular disease area, and again, ensuring that we have strategies that will be able to bring clinical research close to where the patient is. And then also components of measure, ensuring that we're actually have defined measures upfront that we will monitor on an ongoing basis, and that we will make adjustments as needed when we see if particular strategies have been successful or if there's evolving information that we need to shift and change our diversity action plan on the basis of. The guidance also gives us a sense of different approaches that can be taken for implementation of such strategies, and they're by no means sort of exhaustive, but they do highlight the importance of community engagement and partnerships, really deeply engaging with the patient community right up front during trial design, as well as in discussing how to better meet participants throughout their patient journey or their clinical trial journey, and developing partnerships. And these are particularly key to ensure that as we're designing studies and/or strategies, that these truly reflect the community needs, that we think about our eligibility criteria for study to be truly inclusive. And that is really examining each of these criteria, making sure that none will inadvertently exclude certain participant populations, whilst preserving both the ability to determine safety in the course of the study as well as keep study participants safe, that we address any barriers that historically might have been in the place to stop patients not being able to participate in trials, such as the ability to sort of cover transportation to a clinical trial site, childcare needs for participants who have a working day to cover, language support, making sure that our content and materials are both translated into different languages but are culturally sensitive to those languages, as well as potentially examining ways that we can bring trials closer to where patients are without necessarily having them to come to a bricks and mortar site. So thinking about virtualizing, decentralizing, leveraging community care clinics or mobile units, for example. And also, exploring different digital tools and outreach campaigns, again, raising awareness to different participants that will be inclusive and will enable them to consider a clinical trial or clinical research as an option for them in their journey. We also, in terms of the draft guidance, there are regulatory expectations. So this makes it very clear on sponsors what would be required of them to submit a diversity action plan, as we mentioned earlier at the time of the phase 3 pivotal protocol submission. The guidance document also discusses FDA's review process and the process for feedback. Some of the aspects that are not yet covered in the guidance and that many sponsors and others have had some discussions around is, and so how will that feedback be surfaced? What are the ways to address that feedback? But also, what are the consequences of not complying with the expectations of the draft guidance, or indeed putting in efforts that themselves are not successful? What are the discussions and what are the vehicles for having an ongoing dialogue with FDA? And these are some of the areas that sponsors and other trade groups across industry have provided comments on the draft guidance. And if we look at the commitment and the progress to date, one of the things I think it's really important to highlight is that the FDA have had previous draft guidances around diversity plans. This has been a key focus over many years from FDA. And so as sponsors, many sponsors have already been exploring ways to bridge this within their development plan. And at the end of 2024, there was actually a report to Congress, which was part of the Food and Drug Omnibus Reform Act, or FDORA for short. And this report shows the number of diversity plans or diversity action plans that have been received, and also sort of some highlights around whether or not any of these plans and their enrollment goals were met. Again, given that diversity action plans are still a draft guidance, there hasn't actually been a product approved or licensed during the fiscal years with anything that can be reported back on the second topic here. But what we can see is that because of this focus on improving diversity in trials and the new draft guidance in 2024, many sponsors have already been submitting diversity plans or diversity action plans proactively. And so across CEDAR and CBER, FDA has received a number of these from sponsors. And in fact, we can see on the next slide that there have been 139 documents that have been provided in sort of 2023, and there have been 206 that were received in 2024. Although, as we mentioned, the draft guidance is not yet completed and finalized, so we do anticipate that that number will increase when it becomes a formal requirement. However, you can see that across the different divisions of CEDAR, where there's been a large number of these documents across both years that have been submitted, as well as CBER, as well as the device part of the agency. So again, strong commitment from sponsors already to be submitting these documents. And we'll just touch on a couple of aspects of feedback. As the draft guidance is out and has been out on review, and comments have been sent in from many different groups, I think what's been very clear from a large number of these responses is, industry and cross-industry groups really welcome this guidance, welcome both the goals, because we think it's really important to improve representation in clinical research, but also at having that opportunity to comment enables us to sort of help with finalization so that we have a document that's very clear. We all have a goal an intention to address disparities in clinical research, and we also align very strongly with having this document be submitted along with a pivotal study, at which point in time we're typically studying larger groups of patients. We also wanted to acknowledge very clearly, and this webinar is a good illustration of this, that actions truly begin earlier in development. And this guidance document highlights this, but we really want to emphasize this going forward. We're also very clear on what some of the general expectations for a diversity action plan are, based on sort of the inclusions of certain things in the guidance and certain examples, but we would appreciate more examples to illustrate expectations and data presentations, again, to ensure that as sponsors we're meeting the need for data displays. And again, we highlighted that in some cases we know that disparity in healthcare as well as clinical research is a very multifaceted issue, and so it is important that there are different approaches taken to supporting this. Some of those may be more effective than others, and some of achieving enrollment ambitions may not be successful. It's important for us to know what kind of interaction we would have with FDA to be able to explore that when best intentions haven't transpired. Importantly, and this is a sort of something that we'll keep a keen eye on, given that there is also another accompanying guidance around studying sex differences. And, we sort of throughout the document, words like sex and gender have been used interchangeably in some areas. And again, we do think it will be important going forward to engage broader communities around the language and choices of language terms within the guidance. And the final piece I think that we see is a recognition there may be gaps in achieving this in US data and sort of with global trials. How might we describe sort of diverse enrollment across the world from different parts of the world as an adjunct or augmentation to the data gathered within the US for our multi-regional trials. And I think, again, sort of before I hand over to my colleague Beatrice to take us through some very key considerations around how we can think about this in the context of early clinical development in particular, one of the things we also acknowledge that, this is an important guidance to address a lot of historical mistrust in medical research. It's very important that we acknowledge that's also a challenge in being able to achieve our overall aims. And so very critical that all approaches that we take as sponsors and that the agency takes really try and address an improvement in trust in medical research. It's also important that when we work in rare disease trials with smaller patient populations, achieving the full epidemiologic ambitions may not always be feasible with small numbers of patients. And so what are our approaches that we can take there to make sure that the trial is still generalizable and representative of the patients we strive to serve? And also, how do we maintain scientific rigor with the practical implementation of this diversity action plan guidance? So a couple of challenges and considerations as we come into this. But with that, I'll hand over to Beatrice, who's got some excellent points to raise around clinical relevance, but also some of the data-driven approaches in early development.
Thank you, Sean. Now we'll be looking at how diversity affects both the safety and efficacy of drugs and the clinical relevance of the very many variations we have in patient populations. The guidance, it's important to note that it makes reference to many of the different variations. And in fact, the FDA encourages sponsors not only to look at race and ethnicity, but to the many other variables that affect drug safety and efficacy. And in fact, that it should also be included in the diversity action plan. The clinical relevance of this is because there are many contributors to factors that may affect drug metabolism, for example, genetic polymorphisms. There are a lot of variations in receptors and downstream targets that can affect how efficacious a drug is. Differences in body mass and weight can impact the absorption, distribution of a drug, interactions with other drugs, and we certainly know that patients can have concomitant medications which can affect drug safety and efficacy. Other even endogenous substrates like hormones can have influences and are important in sex differences that are noted, as well as environmental factors. Certainly with patient populations, comorbidities can affect drug safety or efficacy, organ function, and tolerance, how long a patient has been exposed to a drug or a class of drugs, can over time affect the safety profile as well as the efficacy profile. Let's examine sex differences, and here are a few case examples of drugs that are influenced by sex differences. And these can be often mediated by variations in body composition, enzyme activity, and hormonal influences between males and females. Zolpidem, for example, females have a decreased clearance, resulting in about 45% higher Cmax and AUCs, which is the area under the concentration curve, which then poses an increased risk for next-day drowsiness and impairment. So it can have significant safety effects. And for these reasons, oftentimes with females, the doses initially will be decreased. Antidepressants also have sex differences. For example, the SSRIs like fluoxetine, paroxetine. Females metabolize some SSRIs more slowly. This can lead to higher drug concentrations and greater responses at lower dose levels. In fact, females are 1.5 times more likely to achieve remission if they're of childbearing potential. And in older women, if they're not on hormonal replacement, they may have poorer responses to antidepressants, which is an important consideration while giving treatment. Other important sex differences are seen with the statins, for example, where females may have a higher risk of statin-associated muscle pain and side effects. Digoxin also is one of the drugs that are adjusted for sex differences, but it's also affected by a host of other factors such as age, renal function, and creatinine clearance, body weight, and concomitant medications. For females, doses are generally decreased to prevent toxicity. This is also the case for individuals with advanced age, low body weight, or impaired renal function, or if they have electrolyte abnormalities that predispose to toxicity. If we look at age differences, warfarin is one example. Coumadin has a very narrow therapeutic range, and there is a risk of major or fatal bleeding when levels are beyond that therapeutic range. And various factors, including age, race, body weight, sex, concomitant medications, and comorbidities, can be important in maintaining or going outside of that therapeutic range. Additional factors such as other drugs and dietary factors, including vitamin K, can affect that therapeutic range and can impose safety implications, as well as genetic factors, CYP2C9 and VKORC1 genotypes. Lower and initial maintenance doses for elderly or debilitated patients and Asian patients are often recommended to ensure that patients are kept within that narrow therapeutic range and are not at greater risk of major or fatal bleeding. One example of a drug that is classified according to racial groups is Bidil, which is isosorbide dinitrate and a combination of hydralazine. This was a heart failure medication that was initially studied in a broad population. When it was submitted to the FDA back in 1997, it was met with an initial rejection. It was determined that the participants in the clinical trials that were submitted were over 70% Caucasian. When a post hoc analysis was conducted, it was determined that the subgroup of patients who were African Americans had significantly better efficacy outcomes. The sponsor then decided to do a confirmatory efficacy study in African Americans in the heart failure trial. And eventually, the FDA approved this particular drug specifically for African American patients. And this is an example of a first time that a drug has been marketed for a specific racial group. Race and ethnicity can also be affected by other classes of drugs. ACE inhibitors, such as lisinopril and enalapril, present a higher risk of angioedema in African Americans and reduced efficacy for controlling hypertension. Warfarin, as we mentioned earlier with the narrow therapeutic index, has more variable dosing in African Americans due to variations in their metabolism, which may increase the risk of bleeding or clotting. Other examples include rosuvastatin, with Crestor, where higher plasma concentration in African Americans can increase the risk of myopathy and kidney problems. There are also glucose 6-phosphate dehydrogenase, or G6PD deficiency-related drug reactions, and these are primarily seen with the antimalarials and some of the antibiotics, such as primaquine and dapsone. The G6PD deficiency is more common in people with African descent, and certain drugs can trigger severe hemolytic anemia. It's important when we look at diversity that we understand that even population subtypes are not homogeneous, and there is a considerable amount of variation within any given group, and diverse drug responses within any given population. So therefore, oftentimes personalized medicine is the clinically relevant approach because it takes into considerations the unique profile of the individual patients and takes into account the wide spread of variables for that patient and how those variables may specifically be affected by a drug in terms of its safety and efficacy. So for example, here is a table showing the prevalence of CYP2D6 and CYP2C19 poor metabolizer phenotypes in various populations. As you can see on the left are various ethnic and racial groups. When we look at the middle column showing the CYP2D6 poor metabolizer status, we see that Caucasians have a higher prevalence of this type of metabolizer status compared to the other populations studied. And you'll note that the prevalence of 7.2 would indicate that the majority of Caucasians may have just regular normal phenotype status or ultra-rapid metabolizer status. And so therefore, there is considerable variability even within a given subgroup. For CYP2C19, we see that the Asian subcontinent, and Polynesian populations have the greatest prevalence of the poor metabolizer phenotype. And again, this is a subset of those populations. So not everyone in those populations will have these poor metabolizer status. However, there's a greater prevalence in these subpopulations. Now we'll talk a little bit about the diversity action plans and how importantly early phase data can inform that plan and help guide good decision making, not only in terms of phase 2, phase 3 trials, but provide meaningful information that's clinically relevant in the prescribing information. As Sean had mentioned earlier, the plans for diversity action are required to be presented no later than the end of phase 2 meeting. which signifies that phase 1 is an important component of addressing some of the variations in safety and efficacy of a drug in development, and will provide very meaningful information to give a more robust evaluation of how diversity affects the drug. We know that phase 1, even though they're limited to small sample size, they present to themselves excellent opportunities to really characterize drug pharmacology. We have the potential to include females with child and non-childbearing potential into these studies, diverse age ranges, racial ethnic group, extensive and poor metabolizers, and patient subgroups. We also will include renal and hepatic impaired studies. These studies offer a perfect opportunity early on in the drug development program to identify relevant characteristics that can affect the absorption, distribution, metabolism, and excretion of the drug, the efficacy profile, the safety, and the efficacy. We're honing in on some of the early proof of concept work, safety, and as well as the potential environmental or drug interactions challenges. And the environmental components can be something, for example, like a food effect study. So there are actually many phase 1 studies that are looking at the surrogates of variances within the populations. And for example, even in vitro and in clinical PK studies, we're looking at sex differences, metabolizer status, and oftentimes phenotyping can be introduced into early phase studies on a more consistent basis to look at some of the variances in response. Drug-drug interaction studies are very important to understand the types of interactions that may occur with the types of drugs that are the most likely to be taken by the patient population that the drug is targeted for. Food interactions are also very important. There can be considerable variations in drug plasma concentrations based on the caloric load or if the drug is administered in a fasted state. This, of course, can be influenced by socioeconomic and an individual's diet. We know that in the United States, we have something that's referred to as food deserts, geographic areas that are very limited in the amount of fresh produce that they have access to. This results oftentimes in diets that are higher in processed and packaged foods, which can affect the daily caloric load of an individual patient and potentially how they metabolize drugs. Ethno-bridging studies are important studies to understand variations in racial ethnic groups. And we also include renal hepatic impairment studies in populations with varying degrees of severity of renal and hepatic disease. Elderly PK studies also provide early phase opportunities to evaluate the pharmacokinetics and potential pharmacodynamic effects of a drug in an older population. All of these types of studies are actually critical for informing the diversity action plan, and not only informing that plan, but also structuring well-designed phase 2, phase 3 studies with more comprehensive dosing paradigms that will allow you to safely dose in diverse patient populations and mitigate any safety concerns that may occur if individuals have certain variables that may lead to increased levels of drug plasma concentrations. There are a number of ways where sponsors and sites such as ours can engage patients to further enhance the diversity of the volunteers that are coming in for studies. One of these ways is by looking at community engagement and partnerships, and that can include partnerships with local organizations, advocacy groups, and healthcare providers that serve diverse populations. Engaging community group leaders to champion trial awareness is a strategy, and holding educational sessions to address concerns about clinical trials, particularly mitigating some of the historical mistrusts around participating in study research. Without mostly the important areas are also to have multilingual materials and culturally appropriate messaging, not only for informed consents, however, but also for the types of measures potentially that are in a trial. And ensuring that the scales and any questionnaires that are being utilized in a study are translated appropriately in a comprehensive fashion. Transparency in research, safety measures, and volunteer rights is very important. And various social media, ethnic media outlets, and patient testimonials can become very helpful in enhancing the engagement. In consideration for recruitment strategies, having inclusive eligibility criteria is important. So there may be some things that you can look at to broaden the inclusion-exclusion criteria to allow for a broader population into the study. So we may be considering things like a broader age range, broader BMI, and of course, all of that has to be done with balancing in mind the safety of the subjects that are involved in the study, making sure that any extensions or any adaptations to inclusion-exclusion criteria are not giving or adding to risk. Reducing barriers to participation, for example, childcare, transportation, language support. Sean had mentioned these kinds of strategies earlier. Can help to ensure that participants are not hindered in their ability to participate in a trial that they would like to take part in. And certainly use of digital tools and outreach campaigns can help to reach out to communities that may otherwise not necessarily be engaged or reached out to for these types of volunteer opportunities. When patients are recruited for a trial, retention is very important. And ensuring that patients are comfortable, have been well informed, have a comfortable environment is very critical to ensuring that they are engaged and retained in the study. You want to have approaches that will reduce volunteer burden, so even study designs to ensure that the criteria and the requirements of the volunteer are not overly burdensome or difficult to accomplish. Accommodating diverse staff is very important, and consistent care across all subjects. It is always important to have transparency with the procedures and study requirements. In fact, that's the fundamentals of the informed consent process. And support for transportation and accessibility are also very important for all those types of studies that we conduct. There are some important questions to ask when planning for the diversity action plan. So based on phase 1 studies, there are important questions that come out that phase 1 studies can help address. First of all, are there notable differences in sex and age? Are there food effects? Does metabolizer status affect the bioavailability? Are there drug interactions that are important and that need to be identified before you move into patient trials that may potentially have exposure to concomitant medications? Are there safety concerns in renal hepatic impaired patients? If any of these questions are asked, there may need to be considerations for dose adjustments or flexible dosing paradigms in the patient studies. Proactive approaches to dose adjustments are important. Not only are those part of the diversity action plan, but those are really critical in establishing appropriate doses for phase 2 and the later subsequent phase 3 trials. There may be a need to reduce doses for patients with a higher bioavailability for a drug, or increase doses that may have suboptimal bioavailability and therefore may need larger doses to achieve an effect or a desired therapeutic target. We always have to remember that subpopulations are not homogeneous, and so it's not a one-size-fits-all approach, as it certainly is based on the individual. So approaches to safety and efficacy must always be individualized to the patient. Assumptions must not be made that a patient will or will not react a certain way simply based on what they may look like or other assumptions. For a strategy, there are additional questions that we must ask, and The first one is, are there relevant pharmacokinetic, safety, and pharmacodynamic variances in the population? And so this is really examining now the target population, patient population for which the drug is indicated for, or the intended indication is for. And are those variances seen in higher prevalences in select population subtypes? And if that's the case, then you may want to focus phase 1 studies examining those variations so that you have an idea of how much of a range you can expect in terms of the safety, the absorption profile, the pharmacokinetics, and all of those parameters that you can measure early on in phase 1 before you start your later phase studies. The critical question is, are dosing adjustments needed in phase 2, phase 3 trials? And if so, this can be easily addressed with approaches of dose titrations and even having early PK sampling in the phase 2, phase 3 trial to mitigate any kind of variances in exposure, particularly if you have a drug like warfarin, for example, that has a very narrow therapeutic index. The other question is, are any groups at higher risk for safety or lack of efficacy? And if so, how can we mitigate those during the phase 2, phase 3 trials and once the drug is approved? This will all serve as very important labeling information so that clinicians can understand how to manage patients safely. So in essence, when you're thinking through a diversity action plan, it really settles on what's clinically relevant to the individual patient, understanding that patients are unique. There are a variety of different variables that can affect the safety and efficacy of a drug. So understanding which patients are most vulnerable to adverse events or lack of efficacy is critical. How are those patients treated and monitored? And how accessible is the drug and associated healthcare? And this last question is more applicable also to the healthcare that's provided during these phase 2, phase 3 studies, but also in the real world once a drug becomes marketed. So the goals of the DAP is to provide ultimately comprehensive drug labeling to identify vulnerable patients, risks, and solutions for dose adjustments and safety monitoring. An ADAPT should inform the above goal. The product label at the end, once a drug is approved, really should be focused on providing clinicians and physicians with adequate information on a proper dose adjustments for different variations of their populations. We also need to ensure that any risks are identified, potential drug interactions, any contraindications, including concomitant medications, as well as any perhaps important safety concerns that could even warrant a black box warning. So all of these together are critical for having a well-informed prescribing information label that will help physicians and patients to safely and effectively use the medication once it's marketed. And of course, we want to always mitigate against unanticipated consequences. One of those could be stereotyping and having a broad expectation, for example, of decreased efficacy or increased risk of side effects in a subgroup. Again, we always have to remember that variations are throughout the populations, and even subgroups have considerable heterogeneity in them. And we can't use a one-size-fits-all approach to medicine. It does require customization to the individual patient. So of course, stereotyping may affect clinical decision-making and access to medications, and so therefore we must undo that. Inappropriate inclusion into clinical trials to meet DAP goals is also another unanticipated consequence or risk. And so, of course, when loosening any inclusion-exclusion criteria to fulfill a DAP goal has to be very carefully examined to ensure that we are not increasing any safety risks in that given patient population or healthy volunteer population for that matter. And so I think that this is really in the context of ensuring really common sense in science and the integrity of the clinical trials that are conducted. So I think that this is really a thorough look at some of the important perspectives of the DAP. Like Sean mentioned, the draft is still in, the guidance is still in draft form, and there may be subsequent changes that are made to it, but it is quite clear from the FDA's footnotes and the guidance itself that it is intended to examine a broader range of these variations that we spoke about in today's webinar, and it must include consideration of those variations. And so I hope you found today's session helpful, and we will now move on to the Q&A portion of today's meeting. If you do have any questions, please feel free to write them into the chat, and we will try and address as many as we can. Thank you. So we have a question that's come in, and it is, how does a sponsor mitigate not meeting the requirements of their diversity plan? Sean, do you have some thoughts on this?
Yeah, sure. And I'm happy to start with a couple of thoughts. And maybe I'll start first and foremost with one of the important parts of this is the ongoing and continual monitoring. It's very important that we continually monitor how we're doing against those sort of enrollment ambitions that we set at the start of the study. That's an opportunity, first and foremost, to adjust and mitigate and put in extra, not waiting till the end of a trial and not achieving those good faith efforts, but really adjusting as we go along. I would also highlight that this is where the community engagement is absolutely key, engaging with the participant community, but also the community where our participants are, as well as insights that our clinical trial sites or different care settings might be bringing, because it could be that there's another barrier that we haven't envisaged that we need to overcome, again, with a different strategy or tactic or approach. And again, so I would just encourage sponsors to monitor on an ongoing basis and make adjustments. I think sometimes as we get deeper into our clinical trial, we may sort of have to make, especially in a global clinical trial, many of the times patients across the world may be able to provide that representativeness overall. And so, again, I would encourage sponsors to think about the diversity of their patient population from around the world that we're receiving data from and how that sort of builds the holistic picture. And that's sort of, again, in sort of very much in keeping with the draft guidance to look at both US displays of data as well as global data, so to think about it holistically. And look at if some of the reason that maybe our original epidemiology estimates may have been either off, we may have to sort of fill in some missing data gaps, we may have done some additional epi work that inform our numbers. And so again, the vehicle of being able to engage with FDA may be adjusting some of the sort of enrollment ambitions during the course of the study. Ultimately, our big aim of all of this is to ensure that we are being representative of the intended patient population, and that's the overarching goal of submitting these diversity action plans, and ensuring that we also make clinical research more accessible to all. So those are some key goals that I view. There may be times where, in order to be able to truly serve our patient population, we may want to sort of enroll faster. And this may mean that there's not always as much time for some of these enrollment approaches to sort of be able to take root. So I think there is, clearly a need for us to have an open discussion and dialogue with FDA in those cases where the greater need of patients is served by having the medicine available, and how we fill those data gaps through the science of the early development, through our science of how a drug would behave across different populations, as well as showing from global studies the representativeness of our data, the intended patient. So those are a couple of thoughts that I have, but it is a reason why many of us have sort of sought those questions so that there can be clarity in those expectations defined up front with the guidance also.
Yeah, absolutely. And I think if a sponsor is finding themselves at the point of an NDA submission and they were not able to fulfill some of the requirements, supplementing, like you mentioned, with some other data points could be beneficial, but also understanding the missing information. If you're doing a sort of a gap analysis of what populations were not adequately represented for whatever reasons, looking at some of the variations, and it's likely that you may have already represented some of those variances in other populations. So if there was an expectation that there may be, for example, a CYP polymorphism in that particular population, you may have already identified other patients that have that particular polymorphism that you can look at and extrapolate some safety and efficacy data from. So there's a lot that can be done with the data set, and also explained and justified. And that was one of your earlier points, was that there's a justification for the inclusion, but there are in cases sometimes where some populations may just not have that condition, or there may not be that as accessible. And so could be a variety of reasons why you may not necessarily meet all of your goals throughout the clinical development program. So being able to extrapolate data and provide a rationale justification for that as part of the NDA, I think would be a good proactive approach before you do your submission, particularly if some of those criteria were not fulfilled in the end. So next question that we have is, how much expansion of the inclusion-exclusion criteria can be implemented to assess the diversity without compromising a study? Well, that's a good question, because that again depends very much on the specific inclusion-exclusion criteria. From an early phase perspective, we tend to have fairly clear inclusion-exclusion criteria for normal healthy subject volunteers. And that's really because the early part of drug development is focused on healthy volunteers that don't have many of the confounders that will sort of muddy the waters for signal interpretation. The goal of earlier drug development in these earlier phase trials is really to understand and characterize the profile of the drug. And so for safety reasons, and particularly in first-in-human studies, you want to ensure that you don't have a population that could have comorbidities or other issues that could compromise our safety. So that really becomes an effort. So in some cases, you may not have as much flexibility, but there are certain provisions that you can include in trials, depending, of course, on the profile of the drug, the safety margin of the drug, the window, and what can be reasonably done without compromising your study design or the safety first and foremost of the subjects. As you get into patient studies, larger studies where you have more room for including different subpopulations and subgroups, you have more information, of course, by that point, is as to the maximum tolerated dose of the drug, what important safety concerns are associated with the drug. You're in a better position to understand what comorbidities, what concomitant medication may be risk factors given in a study design. And so you have some degree of flexibility. And certainly even in early phase studies, we've looked at expanding BMIs, age ranges to include broader populations, even in the early phase trials. But everything has to be done with a lens of course, what is safe to do so. And as you get into these patient trials, we know that there's a lot of diversity in the patient populations, and there will be a lot of comorbidities. Sometimes also for study designs, if there's a certain kind of pathology that's being examined, or if there are confounders, there may be a tendency to exclude certain things because they may have a confounder in the drugs, in the signal of efficacy, or in the safety parameters. So again, these things have to be taken on a case-by-case basis to see how much can be expanded without compromising the objectives of the study and the safety profile. So that, again, it really will depend from the phase of the study, but largely will be guided by the study drug itself and the safety information that comes out, the pharmacokinetic profile, and all of the characteristics of the drug.
And maybe just adding to that, I think, as Beatrice outlined, that's why these early studies are so important, because they really help guide and inform sort of what's safe, and we build the information over time. Ultimately, if the intended patient population who would take this medicine, when it becomes a medicine available to them, if there are a high number of comorbidities with those patient populations, it really is important for us to get to that point that we can broaden our inclusion criteria to encompass those as soon as possible. So working out if it's safe is first and foremost to do so in our early studies. But understanding if that's the patient presentation with the disease, then, then we do have to take very active steps to make sure we study that, whether it's sort of some preclinical models to look proactively to see if it would be safe, or whether it's including small groups in studies earlier in order to understand this. And also just making sure that when we're looking at certain inclusion-exclusion criteria, we're not simply being overly precautionary in the absence of data to suggest an issue. So those are some of the things that we can do as groups, but it has to be done very thoughtfully. And again, patient safety is sort of paramount in all cases, and addressing ways to keep patients safe while still being generalizable to ultimately, the patient condition.
And it's interesting because one of the goals of the guidance is really to also direct prescribing practices and dose adjustments in different in populations, particularly in underrepresented populations or other populations. And we know from the guidance that the racial ethnicity is just one component of many variables that can affect drug safety and efficacy. And very often you see clinical trials in phase 3 all being run at a single dose level, which for certain drugs can be okay. Drugs that have a narrow window, there really does need to be an adjustment. And one of the goals of the guidance, as it's stated, is to determine what, if any, dose adjustments need to be made in populations. And that's a critical component to this, because at the end of the day, it does need to drive prescribing practices, and that information becomes very useful. So if you have populations that have certain metabolic deficiencies, they may have certain characteristics that may make the drug more toxic or less effective or more effective. You want to be able to make reasonable dose adjustments and have those recommendations clearly put out into the label based on these studies. And when these phase 2, phase 3, particularly once you're in phase 3, having that information early on and really understanding the metabolism, the drug-drug interaction effects early on can help guide you on how to best approach dosing in the phase 3 trials, both from whether or not you may need to titrate to effect, which is often a better practice to ensure that each individual patient is monitored for safety and that you can go carefully to the dose that will become efficacious, if it will, and allowing more for flexibility in the protocol design for dosing. Without that, it becomes difficult then to give practical advice on a drug label at the time of the NDA submission once approved on how to do that, unless you're proactively investigating that in your phase 3 trials. And so I think that's also a need for that based on what the guidance is telling us, and based on some across these guidances, not only on race and ethnicity, but also on gender, on pregnancy, on many other subpopulations that are relevant in the patient setting. So this has been a great discussion. Sean, thank you so much for joining. I'm sure we can talk for hours about this topic because there's so many facets to this and how it can be implemented in clinical trials. And we're sorry if we are not able to get to your questions, but we would love to be able to answer them. If you would like to, please use the contact information here to send us your questions. If you've put them into the chat and we've not had a chance to address them, we will by email. So we'll be happy to send our responses to the group at large. And thank you very much for attending today's session. We appreciate you taking the time to spend the afternoon with us discussing a very important topic in clinical drug development. Thank you so much. And thank you, Sean.
Thank you, Beatrice.